Hi, I am Sai Teja. In this video, I am discussing about genetic engineering. What is genetic engineering? Genetic engineering is a technology for modifying the genetic information in a plant, animal, or human in order to produce some desired trait or characteristic. Now we will discuss about techniques of genetic engineering. There are three techniques of genetic engineering: transformation, transduction, recombinant DNA. These are the three techniques of genetic engineering. Now we will discuss about prospects for genetic engineering. There are two prospects for genetic engineering. First one is transgenic engineering. Second one is cloning. What is transgenic engineering? Putting genetic information from one type of plant or animal into another type of plant or animal. What is cloning? Making genetic copies of an existing plant or animal. Now we will discuss about transgenic techniques in different types of organisms. First one, transgenic organisms. An organism is called transgenic if it has genetic information added to it from a different type of organism. By this, humans have begun to do this with plants and animals. Now we will discuss about transgenic techniques in plants. This is the work that is further along. There are examples in transgenic plants, that is corn, soybeans, and cotton, and papayas. These are the examples of transgenic plants. Transgenic crops are being grown in the Americas, South Africa, Europe, Australia, and China. Now we will discuss about transgenic techniques in animals. Human genes have been inserted into bacteria and mice to produce various human proteins for treating various diseases. Now we will discuss about cloning technique. What is cloning? Making genetic copies of an existing plant or animal is called cloning. A clone is a copy of something. In genetics, a clone is a genetic copy of another organism. Clones occur naturally, asexual breeding in plants and lower animals. Now we will discuss about the process of genetic engineering. There are 5 steps involved in this genetic engineering process. First one is the isolation, second one is the cutting, third one is the insertion, fourth one is the transformation, fifth one is the expression. In first step, isolation, this process would take the microorganism and we will isolate the DNA from the subtle microorganism. This process is called isolation in genetic engineering. Second step, cutting. In this step, we will cut the gene of interest from the subtle DNA of microorganism. Third one is insertion or ligation. In this step, we will insert the gene of interest of DNA into the host cell for the transformation. Then we will check for the transformation. In fourth step, transformation. In this step, the insert gene of interest of DNA is transformed into a new organism. In fifth step, the transformed organism is expressed. Now we will discuss about the applications of genetic engineering. Applications of genetic engineering in plants. Weed killer resistant crops. Many types of crop plants have bacterial genes added to them. These genes make the plants resistant to certain weed killers called herbicides. This means that the weed killers kills the weeds but do not affect the transgenic plants. Applications of genetic engineering in animals. There is a growing trend to experiment with inserting protein and secrete it into their milk. This is an example for the applications of genetic engineering in animals. Sheep produce human clotting factor. In this procedure, a human gene has been inserted into the DNA of sheep. By this, it allows the adult sheep to produce a clotting chemical needed by hemophilics to clot their blood. By hemophilics, it is helpful for the clotting of blood. It is produced in the milk of the sheep. This example for the pharmaceuticals. Bacteria make insulin. The human insulin gene has been inserted into a bacterium called E. coli. This allows the bacterium to produce insulin for use of diabetics. In this picture we can see insulin is prepared by a bacteria called E. coli. We will discuss the critical issues of genetic engineering. The release of genetically modified organisms into the environment. Danger of possibility of genetically modified organisms being released into the environment. Genetically modified organisms grown in bioreactors do not provoke as much fear. Genetically modified organisms